This video tutorial is all about DNA profiling. In 1978, Alec Jeffreys was locating tandem repeat sequences of DNA. Tandem repeats are repetitive segments of DNA that do not code for proteins. They may be between 10 and 100 base pairs long, and they only feature the same core sequence: GGG, CAG, GA, XG, where X can be any one of the 4 nucleotide bases. Tandem repeats occur at more than 1,000 locations in the genome, and in each of these places, they may be repeated a random number of times. Some types are highly variable and called variable number tandem repeats, or VNTRs. Jeffreys obtained some DNA from his lab technician and her parents and analysed it. He was surprised to find that the number of tandem repeats showed a family resemblance, but the DNA profile for each family member was unique. He realised that a person's DNA profile could confirm or refute paternity and maternity. There are slightly different procedures for DNA profiling, but the principles underlying them are all sort of following the same 6 steps. So firstly, DNA is obtained from the individual, either by a mouth swab, from saliva on a toothbrush, from blood or hair, or in the case of ancient remains, from bones. The DNA is then digested with restriction enzymes. These enzymes cut the DNA at specific recognition sites. They will cut it into fragments which will vary in size from individual to individual. The fragments are then separated by gel electrophoresis, which we'll learn about in another tutorial. They're stained, and the larger fragments we can see visualized as traveling the shortest distance in the gel. A banding pattern will be seen. The DNA to which the individual is being compared is treated with the same restriction enzyme and also subjected to electrophoresis. The banding pattern of the DNA samples can then be compared. The first method of DNA analysis involved restriction fragment length polymorphism analysis. This method is laborious and is no longer used. Today, short tandem repeats, or STR, sequences of DNA are used. These are highly variable, short repeating lengths of DNA. The exact number of short tandem repeats varies from person to person. STR sequences are separated by gel electrophoresis. Each STR is polymorphic, but the number of alleles in the gene pool for each one is small. 13 STRs are analysed simultaneously. So although STRs, or each STR is present between kind of 5% and 20% of individuals, the chances of 2 people sharing STR sequences at all the loci is 1 × 10 to the 18. This is a greater number than the number of people on Earth. However, there are an estimated 12 million identical twins on Earth. The technique is very sensitive, and even a trace of DNA left when someone touches an object can produce a result. Samples must be treated carefully to avoid contamination. DNA can be stored for many years if a crime case is unresolved. It can then later be used to assess new evidence. We're now going to look at a couple of the applications of DNA profiling. First of all, forensic science. DNA profiling has transformed forensic science. Not only has it brought about convictions and established the innocence of many suspects and of people previously wrongly convicted, it has been used to identify Nazi war criminals hiding in South America, identify the remains of Romanov family and to refute a person's claims to be the survivor. It's been used to identify victims' body parts after air crashes, terrorist attacks, and other disasters, and also match profiles from descendants of those lost during World War I. Another use of DNA profiling is that of maternity and paternity disputes. Half of every child's genetic information comes from the mother and half from the father. Hence, half of the short tandem repeats, the STRs, fragments come from the mother and half from the father. Comparing the DNA profiles of mother, father, and child can therefore establish maternity and/or paternity. Finally, DNA profiling can be used for analysis of disease. Protein electrophoresis can detect the type of hemoglobin present and aid diagnosis of sickle cell anemia. A varying number of repeat sequences for a condition such as Huntington's disease can be detected by electrophoresis, as shown on the slide in front of you.